All right, so thank you for the kind introduction. Switching gears a little bit from MSMS and fragmentation methods to spatial glycomics. And actually not really spatial glycomics, because I'm trying to go towards spatial glycomics and the dynamics of glycosylation in tissue sections via a brief stepping stone, which is some work that we have recently published in Nature Metabolism on the analysis of spatial dynamics of metabolomics on the single cell level, and how we have applied that to reveal altered metabolism after acute kidney injury in, obviously, the kidney. So I didn't do this work on my own. A big team of people have worked on it, and I would like to specifically emphasize the work of Gangqi Wang. He's a very talented postdoctoral researcher in the group of Professor Ton Rabelink, and together we have been pushing this project for quite some time, and happy to get it published recently. So most of the work that I'll be showing you data on today is on direct molecular imaging using MALDI mass spectrometry imaging. And I was actually hoping for Rick Drake to be here to sort of pick out the hot stones for me and explain MALDI imaging, but so I will do it myself, that's fine. So MALDI imaging starts with any kind of fresh frozen or formalin-fixed tissues, which we then section using either a microtome or a cryotome. And then we apply our sample preparation strategy, which in the case of MALDI always incorporates the matrix application. Then we perform spatially correlated mass spectrometry using MALDI, and that gives us a data set in which we can select our analytes and show their intensity distributions in the context of the analyzed tissue morphology. And since this technology is relatively non-destructive to the tissue sections, we can still use the same, the analyzed tissue section for histology staining or even immunofluorescence or immunohistochemistry staining, or we choose to actually extract the remaining molecules from the tissue, perform LC-MS-based analysis, and then do mass matching of our off-tissue analyzed data set with the on-tissue data set to assign molecular identities to the MZ values that we detect in our imaging data set. And as with any mass spectrometry data set, we can then analyze our data in either univariate methods or multivariate methods. And using this same essential technology, we can target different omics layers simply by selecting a different sample preparation procedure. So for example, if we do nothing except for applying MALDI matrix, we can target the metabolome and the lipidome fairly easily. And if we want to make it difficult for ourselves and we want to look at glycomics, we can start from formalin-fixed paraffin-embedded tissues. We apply some tissue washes to get rid of the formalin. We then do sialic acid derivatization to be able to distinguish in tissue between alpha-2,3 and alpha-2,6 sialic acids. We digest our tissues with PNGSF, and then we look at the released glycans in the context of tissue morphology. And then, depending on the mass spectrometer that we use, we can get either high image resolution, which we typically do with MALDI TOF systems. We can go down to subcellular pixel resolution, so 5 micrometer pixels, or we do MALDI FTICR mass spectrometry, and that gives us both the opportunities to do high resolution and high content mass spectrometry imaging. So for the remainder of the talk, I'll be speaking mostly about the metabolomics, the in situ metabolomics using mass spectrometry imaging. And the data that I'll be showing is all recorded in negative ion mode using NEDC. I'll spare you the full name and myself as well, as a MALDI matrix. So a typical spectrum you can see here on the bottom, and on your left side of the spectrum, in the small molecule region, we will find typical secondary metabolites, glycolysis intermediates, TCA cycle intermediates. And on the right side of your spectrum, in the high mass region, we will find the phospholipid, the lipids and the phospholipids that we can detect from these tissues. And as you can see, the phospholipids, they're highly structure specific or cell type specific, and we can use that later on in the protocol that I will show. So ultimately, what we are looking at when we are looking at this type of data is we take a tissue out of an animal or from a patient, and at the moment that we take that sample out, we actually fix the sample, and we're taking a snapshot of the metabolome at that time point for that patient. And obviously, we're now looking at a static image of a very dynamic process, which is the metabolism. Ultimately, what we wanted to do is to show the dynamics of the metabolism in the context of that tissue. And that's why we set out to develop a new protocol, which is the in situ metabolic dynamics workflow that we have published recently, as I said. So we take the fresh kidney from the mouse as starting material, and we use a vibratome to take thick tissue sections, so about 350 micrometers thick, and then these vibratome slices, we will put them in culture medium. And over time, we will start to incorporate isotopically labeled nutrients into the culture medium, and those isotope labeled nutrients are then incorporated into the metabolites, the downstream metabolites of, in this case, glycolysis and TCA cycle. So we chose to work with uniformly labeled glucose, glutamine, and linoleate to be able to target the glycolysis and the TCA cycle metabolism. And then, obviously, we start to see incorporation of the stable isotopes labels into the different metabolites, and with increasing number of cycles, we start to see increasing number of isotope labels being incorporated, and those will all have different masses, so they will pop up in different spots in the mass spectrum, and therefore we can track them over time. So we do that by analyzing all these tissue sections by mass spectrometry imaging. So we quench the tissue culture with liquid nitrogen, we section them, put them on specially coated glass slides, and then we measure them with a MALDI-TOF mass spectrometry. And then we end up with a huge pile of data which we cannot make any real sense of at that time. So we need to integrate all of this information. And to integrate this information, we go to a mathematical strategy that has been developed for the integration of single-cell RNA sequencing data with spatial transcriptomics data. And it's an anchor-based integration, so basically, typically, single-cell RNA sequencing data sets contain about 25,000 genes of information, and the spatial transcriptomics data sets, they contain about 10 to 1,000 genes. However, very likely, a subset of these genes, or maybe all of the spatial transcriptomics analyzed genes, are also going to be present in the single-cell RNA sequencing data set. So they can serve as anchors. So what we do is we take these anchor genes, we do dimensionality reduction, and based on the profile of the anchor genes, we use a Kane nearest neighbor analysis to define which two cells from the two different data sets have the closest matching set of anchor genes. And then we predict, based on these on the anchor genes, we predict the information of the unmeasured genes from the single-cell RNA sequencing data set onto the spatial transcriptomics data set. And this allows us to predict the distribution of unmeasured genes in this spatial transcriptomics data set. And here they did that for spatial transcriptomics data, and they were able to compare then, they were validating the strategy by taking measured genes and look at the predicted patterns by leave one out strategy. So as I said before, the lipid profiles that we detect are very cell type specific, and they always are. We have shown this, others have shown this in the past, and therefore they can serve as anchors between these different data sets to be able to integrate all this data from these different data sets that we have from the different time points and the different nutrients. Provided that these lipid profiles do not change with the introduction of stable isotope labeling, because also the phospholipids that are present in the membrane are products of metabolism, and they can be influenced by the metabolism and therefore also by the incorporation of isotope labeled standards. So that's something we tried to look to find out. So we did all the we basically do spatial segmentation. So we take all our pixels, we perform a UMAP on the total set of pixels, which gives us clusters of pixels that are representing different cell types, and we see that even with the incorporation of stable isotopes at the longest time points that we have measured, these cell type or these UMAP plots remain very specific, and also we don't see notable changes of specific lipid features over time. So therefore we can conclude those lipid profiles are very stable over time, and we can use them as anchors to integrate all of our data sets together. So that's what we then do. And finally, after the integration, we then impute the metabolite information from the labeled isotopomers into the control data sets for each time point. So we have different control data sets for each time point, including all the endogenous metabolite information, as well as all the information from the produced isotopomers for these secondary metabolites that are coming from the cultured tissues. We then have one very big data set with the different time points, with all the different nutrients included. We do, again, a segmentation. We do a spatial mapping, and we do this. We compare the spatial mapping to the post-MSI immunofluorescence that we have performed to accurately identify the different cell type clusters that we have identified, and really try to make sense out of the biology and the morphological context of the tissue. We can then start to calculate metabolic changes over time, so changes in glycolysis, changes in the TCA rate, do pathway convergence, so try to find out which nutrient contributes to which pathway, and all map this back to the morphology of the tissue that we have previously analyzed. And now this method is all very nice, but a method is not really useful if you cannot apply it, so we chose to apply it, since it was a collaboration with the nephrology department, my apologies, to a model for acute kidney injury. And this is the model that is the ischemia-reperfusion injury. So basically, we have taken 3 mice. All of these mice, they had a bilateral ischemia-reperfusion injury surgery, meaning we had one kidney that served as a sham surgery. So basically, we put a clamp on the renal artery, but we didn't close the clamp. And that's the thing we did in the ischemia-reperfusion injury kidney. So basically, the renal artery was closed off for a while, about 18 minutes, and then we took off the clamp from both the sham kidney and the IRI kidney, and then the reperfusion with blood of the kidney causes a lot of damage, and this is particularly a problem when looking at transplantation and transplantation research. Then those mice were allowed to recover for about 2 weeks before sacrificing and taking out the kidneys. And these kidneys were then processed with this entire procedure of vibratome sectioning, tissue culture, and then follow it by mass spectrometry imaging analysis. So now we look at the lipid profiles of those 2 groups of kidneys. So we have the sham group and the IRI group. And basically we find that some clusters are present in both. So we have healthy proximal tubules present in both. We have glomerular cluster of pixels present in both. We have some distal tubules. But then specifically in the IRI group, we see 4 clusters popping up that are unique to the IRI group. So these are pixels that have a molecular phenotype or a molecular pattern that is not present in the sham group. So then we can try to find out which lipids are actually responsible for this offset in clustering. We can visualize those lipids and try to identify them, or at least assign identities to them. And we all found that the lipids that set apart these injury clusters were all oxidized lipids, which is something you would expect if the damage that has been done is mainly caused by oxidative stress. So then the kidney has this feature that it can repair its own proximal tubules after this IRI injury. And that's what we were really interested to look into, to see if we can actually find metabolic differences between different subtypes of this injury. So basically, we did the mass spectrometry analysis. We then did immunofluorescence staining afterwards. So we looked at LTL, which is a marker for proximal tubules. Then we looked at VCAM1, which is a marker for end-stage renal or proximal tubule failure, and KIM1, which is an early marker for early kidney injury. So we extracted all the pixels that were positive for these stainings, and we did, again, a segmentation on this subset of pixels, so to only have information on the proximal tubules and the damaged proximal tubules. And then we, again, get 5 clusters, one with healthy proximal tubules from the S1, S2 segment, healthy proximal tubules from the S3 segment, and then 3 that we were able to correlate to the damaged stainings in the kidney. So now it's very good that we can at least trace these images back to tissue, but we really could have seen this already from the immunofluorescence staining. So we took it a step further and did a pseudo-time analysis to see what the order is of this cluster formation. And then we indeed find that if we start off at a healthy proximal tubule, then we get an intermediate cluster where cells are at least maybe a little bit repaired or maybe not so much damaged. And then we find two clusters, of which the yellow one is the final stage, the end stage cluster where the proximal tubule has completely failed. And we can also perform this trajectory in a spatial context. So we here look at the spatial time in space, let's say. So now this is only looking at lipid profiles, trying to segment our data, trying to make new insights into what different cell populations we have. But we were really interested in the metabolics of it. So since all the information, the metabolic information, is present in the same spectra, we are able to extract it. And we then sought to look at different metabolites and how they are distributed throughout the tissue. So basically, on the next couple of slides, I will only be showing these plots, but this is all that's behind it. So here you see the distribution of the ratio between hexose and +6, so that's the glucose label that we introduced, compared to the total amount of glucose that we have detected. So that's 13C labeled glucose as well as endogenous glucose. And you see that there is a gradient that nicely correlates with previous assignment of these clusters. And if we look at the 13C label enrichment over pseudo-time, we see that there is a decrease of label enrichment over time. And that's basically summarized here. So basically the value that you get here is for each mouse the value of the average value of the blue cluster and the average value of the yellow cluster, and that's what I'll be showing in the next figures. So you have a little bit of an idea where data comes from. So if we then look at the maladaptive repair, so we compare the blue cluster, which is the healthy proximal tubule, with the yellow cluster, end stage, we see that indeed the glucose decreases, also glutamine or glutamate decreases over time. And it seems that lactate seems to be increasing. So this would indicate an increase of glycolysis, but a decrease of TCA cycle activity. We wanted to validate this, so we actually also had a look at the glutamine data set, so where the glutamine was incorporated, and then after the first cycle of the TCA cycle, we have glutamate with five carbons. We see that slightly decreased. And after the second cycle of the TCA cycle, we have glutamate with 3 heavy carbons, and we also see that further decrease, which sort of validated our first observation that the TCA cycle dynamics are downregulated there. So we were able to conclude that maladaptive repair is associated with altered lactate production, so increased glycolysis, decreased TCA cycle. And we were very happy that we were able to show this, because this already was published before, based on spatial transcriptomics data in a similar mouse model, similar study. What was new for us was when we actually started to comparing the metabolic effects of the successful tubule repair. So we have these subpopulations of pixels where we have the healthy proximal tubules in the S1, S2 segment, as well as the healthy proximal tubules in the S3 segment. So based on the staining information, we were not able to distinguish them. They look the same morphologically between the sham mouse and the IRI mouse. So basically, it should all be hunky-dory and it should all be happy and healthy. However, if we looked at the metabolism of these healthy proximal tubules in the IRI mice, we see that pretty much every part of the metabolism is slower, slower in the IRI kidney compared to the sham kidney. And this was something that was never observed before. And basically, this could lead to, for example, this decrease in TCA homeostasis could lead to alterations in energy production and cellular adaptability, as well as could influence epigenetic gene regulation and protein biosynthesis. So now, basically, coming back to why we are all here, and that's glycomics. So this, I said, was going to be a stepping stone towards how we can actually target to look at the spatial dynamics of glycosylation. So Besides that this is only half a figure, you all probably know much better than I do that glycan biosynthesis is dependent on both the glycosyl transferases. I heard today that transcription factors play a major role there as well. And on the other hand, there is the sugar-activated nucleotides, which are byproducts from metabolism or catabolism. And I was really pleased to see that by feeding cells labeled fucose that you can actually track the fucosylation in the cells as well, and that you can find the places where they are located. So what I'm aiming to do now is actually taking a kidney or any other tissue for that matter, label it, perform spatial glycomics and spatial metabolomics, and then do this similar type of data integration method. Because if we choose the right matrix for spatial metabolomics, we can actually see the majority of the UDP or the sugar-activated nucleotides. And we have been doing some cell culture experiments where if we culture the cells long enough, we actually also start to see the labeled versions of the sugar-activated nucleotides in these cells. So I assume that if we can extend this time of culture for the tissues, we will probably start seeing the incorporation in the tissues as well, and potentially even start to see heavy sugars being incorporated into the tissue glycome. And then we can get a grasp on the dynamics of, for example, glycolysis, and how this glycolytic rate can tell you something about the dynamics of glycosylation. So I hope I've been able to show you that the combination of multi-mass spectrometry imaging and isotope tracing is a very powerful combination, and it allows you to track metabolic dynamics in the spatial context of a tissue. On the biology, the failed tubal repair is associated with altered lactate production, and the metabolic deficiencies that we see in recovered proximal tubules between compared to their healthy counterparts. And for the future, we're working to not make this dynamics, but make it properly flux calculations, which is challenging, but we probably will be able to get there. We are currently integrating this with transcriptomics and spatial transcriptomics. Actually, that was published yesterday in Cell Stem Cell. That also involves some other applications, mainly embryology and development of kidney organoids. And then finally, we're going to try and extend this to other omics layers, starting with glycomics. So with that, I would like to thank you all for your attention. Thank the organizers for inviting me to show this little bit of future perspective. And yeah, again, thank you for your attention. If you have questions, let me know.